Okay, so here's just the light reactions again. I want to go over these one more time just to make sure you understand them because it seemed like there was a little bit of confusion still. So photosystem 2, light is captured by the photosystem, funneled down to the central chlorophyll molecules, causes those chlorophyll molecules to lose electrons to the electron transport chain. Those electrons are replaced by electrons coming off of the splitting of water, where oxygen is produced as a byproduct. Electrons flow through the electron transport chain, and as they do, the key thing to the electron transport chain, so the answer to what does the electron transport chain do, well, it pumps hydrogen from the stroma into the thylakoid space, increasing the concentration of hydrogen inside the thylakoid, which sets up membrane potential, sets up a concentration gradient. So hydrogen will now flow through ATP synthase, and that energy of diffusion will drive the synthesis of ATP. So again, if there were holes punctured in the thylakoid, hydrogen could just go through that rather than through ATP synthase. And if it goes through that, no ATP would be produced. So you're going to lower your rate of ATP production as it's going around the turbine rather than through it. Photosystem 1, light hits that. It's captured, funneled down to P700, excites electrons which are sent to NADP reductase to reduce NADP into NADPH. And electrons are replaced in photosystem 1 by electrons coming off the electron transport chain. So in this way, we've created ATP and we've created NADPH. Questions on light reactions before we go forward? Does the answer to that question now make a little bit of sense? So electron flow is not going to be stopped by puncturing holes. Even if the holes were in the middle here, the electrons go from protein to protein, so it's only if you destroy one of those proteins would you stop the flow of electrons. So they don't really interact with the phospholipids. Okay, so today we're going to talk about the next step, and we're going to review this again. I'm going to show you a little video, and then we'll go into one more kind of quirk of photosynthesis. That'll be really it for this chapter. So I introduced this on Monday, but we're going to talk about it in much more depth today. The Calvin cycle, or the carbon reactions. So ATP and NADPH that were produced in the light reactions are now power sources that are going to power the production of carbohydrates in the carbon reactions. So this is the ultimate goal of photosynthesis, is to make sugar, is to make carbohydrates. So the Calvin cycle, the carbon reactions are going to build sugar molecules from smaller carbon molecules, from carbon dioxide, again using the power generated in light reactions. Carbon enters as CO2 and leaves as a 3-carbon sugar named pGal. There's going to be basically 4 names of molecules that I'll expect you to know. One is carbon dioxide, one is pGal, one is RUBP, which we'll get to, and one is something called Rubisco, which we'll get to as well. So the Calvin cycle has 3 stages, and we'll go into each of these stages in a little bit. Not too much detail, but just a little bit of detail about what each of them does. First one is carbon fixation, which uses an enzyme called Rubisco. Second one is pGal synthesis. You're actually going to synthesize the end product in the second step. And then the third step is a regeneration of RUBP. This is a cycle. So a cycle tends to end up where it started, and we're going to see that you're going to end up with the starting material by the end of this, but you are going to produce some pGal, a 3-carbon sugar, every time you go through the Calvin cycle. Okay, so here is the first step, carbon fixation. So, first thing that plants need to do is they need to bring carbon dioxide into the cell. So they do this by taking in carbon dioxide through openings in their leaves, if they are a tree, called stoma. So there's stoma that are on the base of the leaf, where carbon dioxide enters and oxygen gas leaves. CO2 being nonpolar, it can just diffuse right into a cell, and it's going to be attached to a sugar molecule that's already in the cell called RUBP. And this reaction, converting RUBP, which is a 5-carbon sugar, into a 6-carbon intermediate sugar, which I don't expect you to know the name of, so I won't even talk about it, using an enzyme called Rubisco. So Rubisco is a vitally important enzyme. It's what brings carbon dioxide into the Calvin cycle. It's what allows RUBP, a 5-carbon sugar, to become a 6-carbon sugar. So it's a very essential step to happen. You need to be able to pull carbon dioxide in order to make any sugar. So there's actually scientists that spend their entire careers studying Rubisco. Rubisco is so important that it has its own line of research. These are plant physiologists that spend a lot of their time studying the Calvin cycle and studying Rubisco and things that happen to it. Yeah.
Are they forming specific organic compounds?
Yes. I just don't want to have people learn the name. So it's a 6-carbon sugar. So RUBP, a 5-carbon sugar, is converted into a 6-carbon sugar temporarily. All right. So I'm sure if you really want to know it, you can find it out. But it's not something I want to go into in class. So there's actually a lot of steps that I'll leave out of this reaction, a lot of kind of intermediate products. Really the overall picture of it is what I want you to get. If you do go into plant physiology, you'll learn all about all the chemistry of this. So, all right. So carbon fixation is the addition of RUBP or carbon dioxide to RUBP, creating a temporary kind of unstable 6-carbon sugar, and requires Rubisco. Okay. So now that we have this 6-carbon sugar, we can convert it into pGal, which is a 3-carbon sugar. So that second step is pGal synthesis. So the initial 6-carbon organic compound is converted with the aid of ATP and NADPH into 6 molecules of pGal. So we brought in 3 molecules of carbon dioxide, linked it together with 3 5-carbon sugar molecules that were already in there, RUBPs, converted it now into 6 pGal molecules using ATP and NADPH from the light reactions. So at this point, we have 6 copies of our ultimate product, pGal. One of those will leave the cycle, so only one of the 6. The other 5 are going to be converted back into our starting sugar, which was the RUBP again. So pGal synthesis uses energy from the light reactions to make a bunch of pGal, and then after this, the rest of the pGal that doesn't exit the cycle goes back up and forms RUBP again. Yeah.
I'm sorry, you just answered.
Okay. Yeah. So, and that requires a little bit of ATP as well. So this is the regeneration of RUBP. It just rearranges those pGals back into 3 5-carbon sugars, RUBP. So now those RUBPs are all set to combine with 3 more carbon dioxides, be converted again into 6 pGals. 1 will exit, 5 will regenerate RUBP again.
So like it
Okay.
excludes one and then it just recycles the other
Yep,
five?
yep. One leaves and the other 5 are recycled. So, which means, so every time you have 1 pGal exit, how many carbon dioxide molecules do you think need to enter? All right, pGal is a 3-carbon sugar. Maybe that'll be a hint. How many carbons does carbon dioxide have? It only has 1. CO2. CO2. There's only 1 of these. So how many carbon dioxides are needed to have 3 carbons to make sugar? 3 of them. Yeah, 3. So 3 carbon dioxides will enter for every 1 pGal that exits, and the rest of whatever's left over stays in the cycle. Okay, so here's kind of overview of this whole thing. So again, carbon fixation, RUBP that's already present in the cell combines with carbon dioxide. You can see 3 RUBPs, 3 carbon dioxides to make 3 unstable intermediates. And that requires Rubisco to be present. this sugar, a 6-carbon sugar, is going to be converted using energy from the light reactions, ATP and NADPH, to make 6 3-carbon p-gals. One of those will leave. The other 5 will be used to regenerate the RUBPs so that the cycle can continue. And so this will keep going over and over and over again as long as there's energy and as long as there's carbon dioxide. So this p-gal that leaves, this will then be converted through a series of reactions we're not going to talk about into other things like glucose, like fructose, like starch, like cellulose. So all kinds of other carbohydrate products that can be used as either directly to fuel the plant through cellular respiration, or to make storage products like starch, or to actually make physical building components of the cell like cellulose that will be part of the cell wall. Okay. Questions on the Calvin cycle, the carbon reactions? So 3 stages is what I expect you to kind of know a little bit about. And the only...
So it starts with a 5-carbon sugar, right?
Yes, starts with a 5-carbon sugar combining with carbon dioxide. So that's carbon fixation. And then energy is used to make p-gal, that's the p-gal synthesis, and then most of the p-gal regenerates the 5-carbon sugar. That's the regeneration step. And so really the only words you need, only chemicals you need to know are RUBP, Rubisco, and p-gal, and carbon dioxide. That's really it. And then you'll have a pretty good understanding of this. So again, there's a lot of detail left out, but you really don't need that unless you go into plant physiology or plant biochemistry kind of course. Then you might spend a lot of time talking about this because this is hugely important, but I just want you to get the general concept of this. You're going to see a cycle like this in respiration as well. It's a very similar process, different chemicals, but similar idea. Any other questions on this? Okay. All right. So this is a little bit more straightforward than the light reactions. Light reactions had a few more steps to it. I wanted you to know that in a little more detail. Okay. So I have a question for you, just to get you thinking. Okay, a flask containing a photosynthetic alga and a control flask containing just water with no algae are both placed under a bank of lights, which are set to cycle between light and dark, and you're able to measure the dissolved oxygen in both flasks. What do you expect to see? So which of these do you expect to see? So I'll give you a minute to think about this, and then we can talk about it a little bit. Okay, so go ahead and answer if you haven't yet. and we'll talk about this a bit. All right. Everybody answer? All right. Okay, so this is kind of an unfair question in many ways. Partially because we haven't talked about half of the information that you need to answer this, and the other half because there are actually multiple answers that I could make an argument for. But where this comes out of, I actually should have moved this. We used to teach this course, and respiration was before photosynthesis, so you would have already known about cellular respiration at this point. And you really need to know about that to have a better understanding of this. But I just wanted to put this here for us to think about it a little bit today, and I'll probably bring it back again, and we'll discuss it again in the next chapter as well. But from the looks of your answers, at least most of you are thinking in the right direction, which is good. But let's talk a little bit about this. So you have a flask with a photosynthetic organism in them. What's happening to oxygen levels during the day? Is it producing oxygen or consuming it? Producing. Oh, it's okay. So we'll talk about the consuming part later. But where is it producing, or why is it producing oxygen? All right, photosynthesis, right? Well, what part of photosynthesis? So, it's not producing oxygen through respiration. It's producing it through photosynthesis. But what part of photosynthesis produces oxygen? So the light reactions. And why is oxygen produced in the light reactions? Oh. All right, water. What happens to water? Water's split. And why is water split? What's water the source of? Okay. All right, it does produce hydrogen, and it does produce oxygen. Hydrogen's not really it's not immediately needed from it. But what does the light reaction, or what does specifically photosystem 2 take from water? Not energy. No, go to the other one. Electrons, there we go. Yeah, so electrons. So it doesn't take, the energy comes from light. The electrons come from water. And then light energizes those electrons, right? So those electrons, you're right, are going to ultimately power the electron transport chain. But the energy comes from light. Water is the source of electrons. It's the initial source of electrons. It'll be split into hydrogen and oxygen. And that hydrogen, you're right, that does become part of it. It's going to be pumped through the membrane. It's actually going to increase the concentration of hydrogen inside the thylakoid at some point. But the real immediate need for water is to provide electrons. So oxygen is produced as a byproduct of the light reactions. So oxygen levels during the day, they're going to be going up. What happens at night? Is oxygen being produced by plants at night?
No.
Why not?
Because light represents.
So there's no light, so there can't be any light reactions, so there's no need for electrons to feed the light reactions, right?
Yeah.
So that hopefully should be clear. So plants will produce oxygen during the day. They're not going to produce oxygen at night, right? From photosynthesis. So the part of this that you don't know about is respiration. What do you think respiration does to oxygen levels? Reduces them? Is that what you said? Raises them? So when you breathe, do you think you are adding oxygen or you're consuming oxygen? You're consuming. All right, so you know that, right? So your cells don't do photosynthesis, they only do respiration. So what do you think respiration does to oxygen levels? It decreases them, yeah. Yep.
So, wait, is there respiration in plants?
Yep.
So they also use oxygen?
They also use oxygen, yep.
And what part of the cycle do they use it?
The one we haven't talked about yet, which is respiration, which is next chapter. So this is why this is an unfair question, is because I haven't taught you anything about respiration yet. But respiration consumes oxygen. So what do you think's happening to oxygen levels at night then?
They're lower.
All right, so it's not going to stop respiration, so it's not going to stop growing. You know, plants will grow at night. It's not going to stop transporting, making proteins. All the stuff that needs ATP requires respiration, and plants do it just like animals do. They're going to be consuming the sugars that they produce at night, and they're going to be consuming the sugars that they do during the day. So O2 levels will go down in plants at night, or around plants. O2 is also consumed by plants during the day because it's also respiring. So think about this. If I look at total O2 consumed versus total O2 produced by a plant, do you think they're equal, unequal? Which do you think is higher if they're unequal?
It consumes more.
You think it consumes more than it produces? Okay. Anyone think the opposite? Okay. Do you have any evidence that it does one or the other? All right, what do you think? Yeah, okay, so what are you doing right now? You're breathing, you're consuming oxygen. If plants consumed more oxygen than they produced, would there be any left over for us to breathe? No, because we don't produce any oxygen, right? So what do you think they produce the same amount, or they produce more oxygen than they consume? Yeah, they produce way more than they consume. And this is why there's oxygen in our atmosphere. This is why we can breathe oxygen, why non-oxygen producing organisms can exist. This is why we have food to eat. They actually produce way more sugar than they consume because they're producing sugar constantly by photosynthesis and producing oxygen as a byproduct, and then they're only consuming a little bit of it. So they are going to be producing way more oxygen than they consume. So, yeah.
Kind of off topic, but if they would consume everything they produce, does that mean they have like almost a sphere of oxygen around them and everything else would be dead around it?
Yeah, well, yeah, they would be able to, yeah, there would have to be a way that they would not lose that oxygen. It would not go away from them. So it wouldn't be very good for them either because they need oxygen to respire. So they need to not lose that oxygen. But yeah, you know, this is something that we really, I don't know if you, so this is off, it's kind of on the same topic, but you ever heard of biosphere, the biosphere? So it may be before a lot of your time, but it was an idea of a self-contained environment, and they had a bunch of plants that were supposed to produce enough oxygen for the animals to survive, but they found that the levels of CO2 consumption versus O2 production were not in balance. So they were not able to simulate Earth very well. It's a very hard balance to make. Earth does it fantastically. We make way more oxygen by the plants than we consume, and part of it's that a lot of this oxygen production happens at sea. So, but, you know, okay, that's beside the thing. So, knowing that now, which do you think might be true? Okay, so you could make an argument for D that oxygen's going up during the day and it's going down at night, but do you think the oxygen level ever dips below what it started at? No, so you might be able to make an argument for A, that the oxygen's always going to be higher in the plant flask because it's producing way more than it's consuming, so it's going to be keeping on increasing, right? So you can make an argument for A. D would be kind of early on. It might be going up during the day and down at night and up during the day and down at night, but eventually you'd expect it to be up higher, right? I could also make an argument for B, knowing that I'm talking about algae. So algae do what's called blooms. So red tide, you may have heard of that around here. It happens a lot because we have a lot of fertilizer from farms that goes in and gets a lot of nutrients in the water. What algae tend to do is they tend to bloom, which means they'll grow faster than they're actually photosynthesizing, and so they'll be consuming a lot of oxygen at that point. And that's why fish often die, is that the oxygen levels in the water plummet where the algae are, and there's nothing for them to breathe, right? So that could happen as well. But I'd say, you know, so really you could make an argument for A or B or D. C would be kind of hard for it to go right back down to that baseline level every time. And it's probably not going to be E, or it's definitely not going to be E. But you can make an argument for a lot of them, right? So all of you were right, because no one chose E, right? But there you go. It's hard to say. You know, the more I think about this, the more I kind of agree with A. That definitely after a few cycles of this, oxygen's probably going to be higher in the flask at all times with the algae. But, you know, it's hard to say. So really, this is not a great question, but it's a kind of discussion question, right? Okay. This flask. So if this was. No. A flask does not have to contain alcohol. Yeah. Okay. So I'm going to show you a video, and then we're going to have a little bit of time to review, and then I'm going to talk about desert plants.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
So that's a pretty good video. So that'll be part of the podcast for today. So if you want to watch that again. I really like the way they've kind of summarized everything. They do use some different words, so they say G3P in the end rather than PGAL. We're going to use the one from your textbook so that it's easier. So you look at different texts, they're all going to use different terms for these things. So any questions on the light reactions or the carbon cycle? Okay. Those steps make sense for the most part. So the light reactions produce ATP, they produce NADPH. by capturing light energy, using it to energize electrons, and those electrons then produce this one through concentration gradient, right? And then those energetic products go to the Calvin cycle or the carbon cycle to take carbon dioxide and convert it through a series of reactions into pGal. So, okay. So, photosynthesis, very, very important process. So, it is the process by which light energy is converted into chemical energy of organic compounds. So, it's how all sugar is produced. So, we would not be able to survive without it. It provides us with energy sources ultimately, either through us eating plants or for us eating things that eat plants. So sugar made in the chloroplast through these processes of photosynthesis will supply chemical energy, so in the form of actually sugars that can be broken down through the process we're talking about in the next chapter, respiration, into ATP. And it also provides carbon skeletons to synthesize organic molecules of the cells. So it provides structural components. So cellulose in plants is made by combining those sugars in a different way to actually make the cell wall and to allow plants to have a structure to them. And then, animals as well will use these products to either as a fuel source or as a structural component. So things like chitin are made from sugars as well. Plants will store their excess sugar as starches in structures such as roots, tubers, seeds, and fruits. And this is why these are the things we like to eat. So we like to eat carrots, which is a root. We like to eat potatoes, which is a tuber. We like to eat seeds and fruits as well, which are seeds and fruits. So they provide that's the reason why we like these is that they're filled with carbohydrates, that they're very tasty because they provide us with lots of energy. And in addition to producing foods that are really for storage of excess material for the plants so they can survive off of that later, they also provide oxygen as a byproduct, which is a necessary component for cell respiration, which we'll learn about starting on Friday. Okay. All right, so photosynthesis, process that feeds the earth. This is why this is a really kind of important topic to understand, because this is how you can exist. So there's one more little step to this chapter, one little extra little bit. It's kind of an interesting kind of addendum that's talking about these plants called C4 plants and CAM plants. And I don't particularly expect you to know the intricate details of C4 and CAM, even though I'm going to talk about them right now. Mostly I want you to know the overall picture of what the advantages and the disadvantages to being a C4 or a CAM plant. So the Calvin cycle is known as a C3 pathway. So the carbon cycle that we just talked about is a C3 pathway because it makes a 3-carbon sugar by the end. So C3, 3 carbons. Right? The Calvin cycle gets its carbon supply by CO2 diffusing into the cell and originally coming into the leaf of a plant, most land plants, through stoma. So a stoma is an opening in the bottom side of most leaves that allows gas exchange, allows carbon dioxide in, and allows oxygen to leave. It also, though, is a source of water loss for plants. So plants will have water evaporate out of the stoma, and that is one method of pulling water up from the roots. However, if it's a hot, dry day where there's not much water in the ground, plants don't want to lose that water. They don't want that water to evaporate out of their leaves because, as we've seen, water is an essential source of electrons for the light reactions, and without water, you wouldn't have the light reactions functioning, you wouldn't be producing sugars, you won't survive. So on hot, dry days, plants will close their stoma, which will prevent water loss, but also inhibits gas exchange. So if they're not bringing carbon dioxide in and they're not releasing oxygen, carbon dioxide levels inside the plant are going to go down, and oxygen levels inside the plant, if they keep doing photosynthesis, are going to go up. So this causes something weird to happen. So Rubisco, though Rubisco is really good at catalyzing that carbon fixation reaction of adding carbon dioxide to RUBP, when oxygen levels are high, when O2 is high inside the plant, Rubisco, instead of adding CO2, it adds oxygen to RUBP. This is a process called photorespiration, and photorespiration actually causes a breakdown in the sugar molecules that are inside the plant. So some plants on really hot, dry days can lose 50 to 80% of the sugars inside that they've already created. So instead of photosynthesis making sugar, it's actually consuming sugar. That's what's called photorespiration. And it doesn't produce any ATP, it doesn't produce any NADPH, it just consumes sugar and doesn't produce anything. So a really bad thing. And this is really a relic of the kind of evolution of Rubisco. Rubisco first appeared when there was very little oxygen in the atmosphere, so it didn't have to worry about oxygen. So it was kind of, it's got this inefficient process. So photorespiration wastes ATP and NADPH, and it doesn't create any sugar. So plants that typically live in hot, dry environments, so desert-like environments, need a way to deal with this. They need a way to prevent photorespiration from occurring. So we're going to talk about two ways that this occurs, the C4 pathway and then the CAM pathway. So C4 plants, they have this different arrangement of cells inside of their leaves. They have two distinct types of cells, one called mesophyll cells here, and then kind of on the inside near the veins, which will eventually transport the sugars, are bundle sheath cells. And what they do is they separate the carbon reactions. They separate the carbon reactions from the light reactions. And they'll do the light reactions in the mesophyll cells, and they'll actually do carbon fixation there as well. And they'll do the Calvin cycle, the carbon reactions, in the bundle sheath cell. So where they produce the oxygen is separate from where they consume the carbon dioxide. So here's what it looks like in a cross-section here of a C4 plant. So here is the mesophyll cell on the top, here's the bundle sheath cell here, and here's the vascular tissue. This is where it will eventually transport the sugar. So light reactions and carbon fixation occurs in a mesophyll cell. So oxygen is produced there. This is where oxygen levels will be the highest. This is where carbon dioxide will be the lowest. And what it does is instead of using Rubisco, which will pull in oxygen if oxygen levels are high, it uses something called PEP carboxylase to initially take carbon dioxide in and convert it into a 4-carbon compound, this oxyl acetate here, which will then be transported to the bundle sheath cell where it will release carbon dioxide, and Rubisco will pick it up and do the Calvin cycle. So it's got an initial step that it makes a 4-carbon sugar, and this is why it's called the C4 pathway, because it makes a 4-carbon sugar initially. So oxygen levels in the bundle sheath cell never go up, because it keeps the oxygen in that mesophyll cell, and carbon dioxide levels always stay pretty high because it's funneling carbon dioxide into the bundle sheath cell. PEP carboxylase is much more efficient at pulling carbon dioxide out of low carbon dioxide, high oxygen environments. But there's a downside. So the downside of this is that this initial C4 cycle requires ATP. Okay. So if you grow a C3 plant next to a C4 plant and you provide them with plenty of water and equal amounts of sunlight, which do you think is going to grow faster? Yeah, C3. Why the C3? That's right. Yeah, because it doesn't need to burn this extra ATP. So per photon that hits it in the light reactions, the C3 plant is going to make a little bit more sugar than the C4 plant. So grown next to each other, C3 plant grows faster. But what if you take both of those plants and put them in the desert? What do you think will happen to the C3 plant? C3 plant will die. C4 plant will survive, right? So it's a trade-off between surviving in hot, dry climates and being efficient at growing. So a little bit less efficient. And this is why all plants don't do this. So plants that grow around here, they don't need this. We tend to have our hot days when it's really wet, and we tend to have our dry days when it's not so hot, right? So the winter, it's not as hot, but it's dry. And in the summer, when it's really, really hot, it tends to be swampy, right? So we have plenty of water usually. Okay, so that's one method. Here's the other method, which is similar idea, but just a different way. So in CAM plants, crassulacean acid metabolism, which you don't need to know that name, but just be able to recognize CAM, instead of having two separate cells, one for carbon fixation and light reactions and one for the Calvin cycle, they have one cell, but they fix carbon at night. So they open up their stoma at night and fix carbon into an organic acid called malate. They then feed malate to the Calvin cycle during the day so that they don't have a decrease in carbon dioxide levels. Rubisco will continue to add carbon dioxide rather than oxygen, and they're going to do fine. And they only have their stoma open at night, so they're not going to be losing water during the day. So a lot of desert plants are like this. The nighttime, it tends to be cooler. There's not as much sun energy to cause evaporation. They can open their stoma and pull in carbon dioxide all night long. So they'll fix a ton of carbon dioxide at night and then feed the Calvin cycle during the day. Okay, so CAM plants, night versus day, whereas C4 plants have two different cells, one for fixing carbon and light reactions and one for the Calvin cycle.
Can plants like adapt if you put
No, they're going to do one or the other. So plants are either C3, C4, or CAM.
What's the one that blooms at night? Isn't there like a flower or something that
Yeah,
only
and that's a different thing. That's for reproduction, right? So flowers are for reproduction, but it's the same kind of concept, is that they're targeting pollinators that are only active at night. So they're only going to put that energy forth at night to do that. So, but yeah, that's kind of a different thing. It's not for photosynthesis, it's for reproduction. Okay, so just to summarize, both the C4 and the CAM pathways require extra ATP. So again, that CAM pathway to fix that organic, or to fix that acid at night requires ATP. Therefore, they're less efficient than a C3 plant, yet they can survive in hot, dry climates. So if you grow a cactus next to a vine, if you water them both, the vine's going to grow a lot faster, but if you stop watering them for 2 months, the vine will die, the cactus will survive, right? So cactus, really good for people that don't take care of their plants, but it grows very slowly, so don't sit and watch it grow, right? So that's the end of the photosynthesis chapter. Review all this material, I guess, and hopefully you've already started in your homework. Friday, we're going to start in the next chapter, which is respiration, so you'll learn the second half of that question that we went over. All right, that's it.